of all, hello. I'm quite happy to be here. I'm originally from South America, so even if I'm now in France, this is it's nice to go back a little bit to speak with people from my region. And now I'm basically going to present some of the lines of the MIND team, which is in INRIA-Brasil, mostly the lines of work that I do. And the idea is to link from neuroimaging to cognition. Yeah. So basically, the point is why we want neuroimaging to go beyond radiology. Yeah, at some point, MRI was used only for diagnosis, which is mostly based on the idea of we see an image, and then from that we get a diagnosis. But our idea is to use neuroimaging to understand cognition. And if we can understand cognition, we can also understand disease and get impact on electronic health. And we have seen already, from a long time ago, these points were like this old Nature paper where they show that you can detect changes in the gray matter after cognitive training. So basically, the way of understanding cognition from neuroimages has been paid from a long time. And what is happening now is that in the recent years, the sizes of the data sets that we can use to actually train these algorithms and use AI tools have become quite significant, even if in the previous presentation by Edson Amaro, he was saying how there's a lot of biases in this that, of course, we should campaign to fix. But still, with these biases, we can show that this provides a window into how the brain works and hence enhances the point of having a more diverse population, which was Edson's point. Yep, Edison, sorry. So I'm going to take a machine learning viewpoint to this, which is basically I'm going to consider MRI modalities as the data and all of the treatment that we do generally in functional MRI, diffusion MRI, or structural MRI as a feature extraction. And then I'm going to just consider these data features, and our main goal would be to do what we call a downstream task, which is, for instance, trying to predict either diagnosis, like whether a person is a normal controller or it's actually ailing from schizophrenia, or whether we can predict the cognitive performance of a certain subject. And then once we have actually established this idea of that we can predict behavior from these features or predict diagnosis from these features, then we can go to this idea of feature importance and see which of the features were the most important to actually provide us with this either classification in terms of disease or try to predict cognitive ability. So an example of feature extraction will be functional connectivity, where we have, for instance, resting state fMRI on the left, and from there we go to the time series on different regions that we can actually compute in terms of their correlation with a specific area of the brain, like the visual word form area. And this was work that we did with my colleagues in Stanford, and the idea at the end was basically whether from these features we could predict cognition. And other set of features that we see, for instance, in diffusion MRI can be the quantification of microstructure, where basically on the left we have one of the initial tasks of trying to see if, looking at the diffusion MRI signal, we could see if a water particle was in a free environment, such as the ventricles, or it was in a restricted environment, such as within the brain gray matter or a neuron. And on the right side of that slide, we see basically large features of the whole brain, like tractography, which actually try to extract from diffusion the connections between two parts of the brain. Now, the way that this went for a bit is that once we had these features, we will try to examine these features. We will try to do a downstream task. And generally, the downstream task for many years was basically based on t-tests. For instance, here by Vinod Menon, also we have one of the first moments where they try to detect which region in the brain was actually reacting to music. And for that, the downstream task was basically I'm playing to a person music or scrambled music and try to see whether there was a differentiable activation in the brain in an area that was more responsive to some sort of sorted musical score more than just random noises. In terms of diffusion MRI, one of the first downstream tasks was this idea of trying to differentiate on the left, for instance, changes in the optical nerve based on whether you had neuritis. Yeah, and on the right, we're trying to detect, for instance, changes characteristics from schizophrenia. And if you see what's happening here, the samples at this point, which is sort of 20 years ago, were extremely small. And so the main concept was to use hypothesis testing, which has been designed at the beginning of the 20th century to basically act on very small samples of very clean data. And from here, I'm going to go to a little digression where the traditional procedure is to get a phenomenon, model it, yeah? For instance, extract a feature from the language network or from the return to origin probability in terms of diffusion, fit this model to the data, and then try to reject the fact that this model hasn't actually fit a null hypothesis of not being related with the data. And at least to me, the overuse of hypothesis testing like this is at the epicenter of the reproducibility crisis that we have in all of these areas today. So we're trying to migrate to an idea of predictive modeling, which is basically harnessing the large data sets that exist, and from the model feeding, then trying to predict data. And from this prediction, we can use holdout data sets that can tell us basically whether a prediction is actually capturing the phenomenon. Once we can tabulate whether our prediction is good, which is something that's much more common, for instance, in the machine learning areas than it would be in the cognitive science labs, at least in general, at the current state of science, we can argue in a more reproducible way whether we are capturing a phenomenon through what we call a downstream task. So, for instance, one of these examples that we have is we developed a feature extraction algorithm with a student, Rutger Fick, which was basically trying to get from the diffusion signal. I don't know if you see my pointer. Yes, I'm guessing you do. We did extract not only the orientation functions from where like the principal areas of diffusion, but we're also trying to extract some sort of quantitative measurement that's related to whether the water particles in the brain are moving inside of a restricted or a non-restricted environment, and basically something which is reacting with respect to the volume of the restriction. For instance, if we are in an area that's plagued with or that's filled with neurons, we can try to see if we have all the conditions whether the sizes of the neurons are different across different areas, provided that the conditions are there. So once we developed this, what did we do? We went and we analyzed the human insula. And the human insula, it's quite interesting because it has, as you see here from histology, an area where you have very, very large neurons and some sort of diagonal angle from the frontal ventral to the dorsoposterior regions, where there's very large neurons here and very small neurons here. So knowing that this phenomenon existed, we tried to look with RTOP, which basically quantifies the time that a water particle in average takes to get back to its origin, whether here we will find very large areas, very large neurons, or a measurement that could be attributed to very large neurons, and a measurement that could be attributed to very small neurons, more on the posterior side. So now we have a way to extract features from the signal and some sort of qualitative assessment that we are capturing a cellular phenomena in the human cortex. Then we took it one step further and we tried to predict from this cognitive abilities. So basically we did a multivariate analysis where we fit weights of cognitive abilities related to cognitive control, such as sorting cards, for instance. And this is basically our fitting with respect to the measurements of RTOP in the insula. And we can see that the fitting is non-negligible, but again, trying to attribute cognition to only one phenomenon in the brain, which is actually static or of like slow change, like neuronal, like sizes or distributions of neurons, it's quite limited. So 30% here, it's quite interesting. But more interesting to us is we were able to predict with a correlation explaining up to 20% of the variance here, we were able to predict cognitive ability. So basically, we have now, we can go from feature extraction to some qualitative hypothesis about measuring a phenomena, and then use these features to predict to predict cognition, which means that at least for us, it meant that we were capturing interesting information about the brain. And again, our main idea here is to validate these measurements or these techniques through the prediction of cognition or some out-of-scanner score. Basically, that was our conclusion. And we kept with this idea. So for instance, we have what we have here is the connectivity that we can extract through tractography, which is a procedure to extract white matter tracks from a diffusion magnetic resonance image that then can be represented as a connectivity system, which means that here we have a higher color when it's much more likely that from the diffusion imaging, we got evidence that these two areas were connected. So now the question is, do these imaging characteristics actually correlate with a physical phenomena? So to measure that, we went into an intrasurgical, into set of surgical operations with our postdoc, Patrick Philippiac. And basically what you have here is a section of the brain that you can see here in the window. Here it's the frontal part, here's more the posterior temporal part, where we dispose a set of electrodes and then we stimulate it in different areas. And the idea here is that if we're stimulating in these areas, we should see communication in the electrodes that you see here in red. And if we can predict a phenomenon, an electrical phenomenon of the stimulation from the connectivity, like for instance here, the time that we get to the first negative voltage peak, it means that somehow we are capturing something of the physiology with the diffusion imaging. And what you see here is that by using the number of fibers in the tractography and microstructure measurements like we used for RTOP before, for the insula before, we can, in a biased way, predict this delay. With here a Blantalmont plot where we show here the 0 means that perfect prediction, here it means that we over predicted the delay, and here that we under predicted it. Yeah. And what you see is that in general, our standard deviation is on about 10 milliseconds, which it's actually quite good for the phenomena. And even if we have a biased algorithm, which means over prediction on the low scores and under prediction on the high scores, we are capturing some information that can be related from MRI to a phenomenon which is completely outside of the MRI machine. And this is all through predictive analysis, basically fitting a machine learning model to the features obtained by diffusion, training them with the delays of a set of subjects, and then try to see if we can predict it in unseen subjects, which was actually our case. And then taking this one step further, in our Nature Communications paper published with spearheaded by Lang Chen, we used these with these measurements of connectivity to predict cognitive abilities such as picture vocabulary or word reading from the structural connectivity of the area of regions connected with the visual word form area, which actually enabled us to see that with the structural connectivity, we could predict quite a small amount of the ability of word reading, but still there's something there to push us to try to get better imaging and keep exploring this idea of predictive cognition from structural connectivity, with the idea that in time, if we can predict cognition in the healthy brain, we can also, for instance, predict cognitive decay in certain pathologies like multiple sclerosis. So we could predict some performance, and it was a low percentage, but still now we are trying to get to multimodal approaches and trying to make it a little bit more complete. Sorry, yeah, I already said this. And finally, the thing is whether it makes sense to actually study feature importance after this. What does it mean whether it makes sense is, can we actually use these features to extract information? And in works that were with Victoria Shevchenko that was actually approved in our journal for diagnosis, or with the actual article from Lang Chen, we can actually then invert this prediction and try to scramble the prediction and get which areas are more important for one task or the other. And what we could see here was that, for instance, the temporal connections of the VWFA were more important for reading, and other tasks were actually for the dorsal ventral stream here. So basically, we could split the tasks of the VWFA into tasks relating to reading and tasks relating to cognitive control. With respect to Victoria Speshenko, we were able to show that the central sulcus and the visual area were actually important markers in trying to determine the diagnosis of schizophrenia. We still have not gone into interpretations of that, but from a feature importance perspective, just pure machine learning, it seems to be interesting to see that there are some interesting extractions of features there from, and I forgot to say it, from resting state fMRI. So basically, the person is doing absolutely nothing in the MRI scanner here, and we're taking fMRI images. Sorry, I'm going to, this I was meant to skip because we don't have a lot of time. So I'm sorry.
Just 2 minutes. Thank you.
What? Sorry?
Could you finish in 2 minutes so we can have questions?
Yes, that's why I'm skipping this part.
Thank you.
Yeah. Yeah. So, and my last part, it's with about trying to get feature extraction at the individual level from fMRI. And here what we have is a model where we actually model an fMRI input in terms of one subject's acquisition of the fMRI connectivity and a group fMRI connectivity. And on the other side, what we do is we model the brain divided into networks, which is here it's a hard division. Then we have a soft division. So basically, what's the likelihood that one of these points in the brain belong to a network, and here at the individual level and here at the group level. And our main idea here is that by getting an individualized division of the brain, we can get better prediction of cognitive tasks. The main point here is that if we take a database of 1,000 subjects with 60,000 points in the brain, we have 60 million observations, which is basically hundreds of millions of random variables to actually fit this model, which actually we're able to develop a technology to fit. And it's in our recent publications in ICLR. And the point here is that we're using a combination of generative modeling and deep learning to solve it. Why? Because the idea is that deep learning networks are really, really good for regression and for trying to fit and predict data, but they have not been developed to explain why this prediction is happening or to detect features in the brain or why we can predict cognition. So then this is why we went from this generative modeling where we are actually making a big hypothesis about the structure, which is and then go into the deep learning system to solve it. I'm not going to get into the technical details of how we derive automatically from the generative modeling a machine learning network based on deep learning to solve it because of time. But what I can tell you is that we obtained individual parcellations. Then we were able to get a set of cognitive tasks and find for those parcellations a regression. And we were able to, with different features like the parcellation or the connectivity, we're able to predict in average 20% of cognition, which is up to the state of the art today in prediction of cognition. And finally, my slides, my couple of final slides is that the problem of this is that this needs thousands of subjects to train the model. And this is generally not feasible in clinical data sets because clinical data sets are, if lucky, of 100 subjects if you have someone who's keeping very good record, at least with the hospitals I've been working in France. So what we did is with this modeling, we were able to pre-train this model in a large data set like HCP, and then predicting a small data set like CAMCAN, which is completely different acquisition parameters for the same resting state fMRI. And what you can see here is what is observable here is that by priming our system with the pre-trained model, we can boost basically our prediction by almost like getting to 20% of explained variance when we had 5% without priming it with a large data set. So the conclusion from this is that we can actually use a large data set to train a model and then transfer this training into a small data set, even if the acquisition parameters are worse, and gain on the cognitive prediction, which our hope is will enable the use of predictive modeling through AI in clinical settings where large data sets are not available. And with that, I thank you for the invitation here. I want to thank all my different collaborators and funding sources, and I will be happy to receive your questions.
Thank you, Demian.